What if I told you that aging is not what your doctor thinks it is? What if the gray hair, the slowing metabolism, the creaking joints, the fading memory, what if none of that was inevitable? And what if the scientific community has already proven in peer-reviewed laboratories at the world's most prestigious research institutions that the biological clock inside every one of your cells can be slowed and in measurable documented ways actually reversed? Not with a drug, not with a procedure, with specific, accessible, evidence-supported interventions that most people over 60 have never been told about in a single medical appointment. I'm Dr. William Lee, and I have spent 30 years at the intersection of nutrition science, vascular biology, and the emerging field of longevity research. What I am sharing with you today is not speculation. It is not wellness culture optimism. It is published science, and the implications of that science for every man and woman over 60 watching this video are profound enough that I consider it a professional obligation to bring it to you directly. A study published in the journal Nature Aging by researchers at Harvard Medical School and the University of California, San Diego, demonstrated that specific dietary and lifestyle interventions reversed biological age measured through a highly validated tool called the DNA methylation clock, which I will explain momentarily, by an average of 3.23 years in just 8 weeks. 8 weeks. A study from the Salk Institute published in the same journal showed that partial cellular reprogramming using naturally occurring biological signals reversed aging markers in living tissue by up to 40%. These are not fringe findings. These are among the most cited papers in modern biomedical research, and they point toward the same conclusion. Aging at the cellular level is far more malleable than medicine has historically acknowledged. Before I reveal the most extraordinary item on today's countdown, and I can tell you that number one involves a mechanism so fundamental to your DNA that pharmaceutical companies have spent billions of dollars trying to replicate it artificially when the natural version has been sitting in grocery stores for decades, I want to hear from you right now. Leave a comment below telling me your age and what concerns you most about aging. Your memory, your energy, your physical strength, or something else entirely. I read every single comment on this channel, and your answer shapes the content I bring you every week. Now, we are counting down 6 scientifically proven interventions that repair DNA, slow biological aging, and in documented cases, reverse it, ranked from powerful to the most extraordinary finding in longevity science today. At number 6, and this is the entry point to understanding everything that follows a concept that rewrites what most people believe aging fundamentally is: the protection and lengthening of your telomeres, the biological timekeepers sitting at the end of every strand of DNA in your body. Telomeres are the caps at the end of your chromosomes. Think of them exactly like the plastic tips at the end of a shoelace. Every time a cell divides to replace itself, those plastic tips get slightly shorter. When they become too short, the cell can no longer divide safely. It either dies or becomes what scientists call senescent, a zombie state where the cell stops functioning but refuses to die and instead releases inflammatory chemicals that damage every surrounding healthy cell. This process of telomere shortening is one of the most fundamental, well-documented mechanisms of biological aging. And here is what your doctor almost certainly has never told you directly. Telomere length is not fixed. It is not simply determined by genetics and time. It is profoundly modifiable by specific behaviors, and the enzyme responsible for rebuilding telomeres, called telomerase, can be activated by dietary and lifestyle interventions that have been proven in human clinical trials. Research from the Preventive Medicine Research Institute, led by Dr. Dean Ornish in collaboration with the University of California, San Francisco, conducted a landmark 5-year study following men who adopted a comprehensive lifestyle intervention, including specific dietary changes, stress management, and moderate exercise. The result was the first human study ever to demonstrate that lifestyle changes actually increased telomerase activity by 29% and lengthened telomeres over time. This study was published in The Lancet Oncology, one of the most rigorous medical journals in the world. The control group, who made no changes, continued to show telomere shortening consistent with normal biological aging. After the age of 60, telomere shortening accelerates by approximately 1.3 times compared to middle age, according to research from King's College London. This acceleration is driven by oxidative stress, the cellular equivalent of rust and chronic inflammation, both of which increase dramatically in the aging body due to declining antioxidant defenses and the loss of hormonal protection. The interventions that most powerfully slow and reverse telomere loss all share a common mechanism. They reduce oxidative stress and inflammation at the cellular level while simultaneously stimulating telomerase, the rebuilding enzyme. The most evidence-supported dietary telomere protectors are omega-3 fatty acids from wild-caught marine sources, with a study from the University of California, San Francisco, showing that each standard deviation increase in omega-3 levels was associated with a 32% reduction in telomere shortening rate over 5 years. Polyphenol-rich foods, including dark berries, extra virgin olive oil, and green tea, and the mineral zinc, which is a direct cofactor for telomerase enzyme function and is deficient in an estimated 35% of adults over 65. For practical implementation, 2 servings of wild-caught fatty fish per week, 1 tablespoon of extra virgin olive oil daily, 1 cup of mixed dark berries daily, and a zinc-rich food pumpkin seeds, legumes, or lean red meat 3 to 4 times per week. For synergy, consume your omega-3 source alongside vitamin D, as research from Ohio State University found that the combination of omega-3 and vitamin D3 was 40% more protective of telomere length than either nutrient alone, likely because both compounds influence the same inflammatory gene expression pathways that drive telomere erosion. Number 5. And this is where the science shifts from protecting what you have to actively repairing damage that has already occurred, something that, until very recently, was considered biologically impossible. The activation of your body's own DNA repair enzymes through specific nutritional compounds that most people over 60 are profoundly deficient in. Every single day, the DNA in each of your roughly 37 trillion cells sustains between 10,000 and 100,000 molecular lesions, damage events caused by radiation, oxidative stress, metabolic byproducts, and environmental exposures. In a young, healthy body, a sophisticated network of DNA repair enzymes moves through the genome continuously, finding these lesions and correcting them with extraordinary precision. Think of these enzymes as an army of microscopic editors constantly proofreading your genetic code and correcting every typo before it becomes permanent. When this repair system works efficiently, damaged DNA is corrected before it can cause cellular dysfunction, mutation, or the senescence cascade that accelerates aging. After the age of 60, DNA repair enzyme activity declines by approximately 30 to 50%, depending on the specific repair pathway, according to research from the National Institute on Aging. The editors are still there, but they are working at half speed, missing errors they would once have caught instantly. The accumulating DNA damage that results is a primary driver of cellular aging, cancer risk elevation, and the functional decline that most people attribute simply to getting older. The most powerful naturally occurring activators of DNA repair enzymes are a class of compounds called NAD+ precursors, and I want to explain NAD+ clearly because it is one of the most important molecules in longevity science and one of the most misunderstood. NAD+ stands for nicotinamide adenine dinucleotide. Think of it as the electrical current that powers your DNA repair machinery. Without adequate NAD+, your repair enzymes called PARPs and sirtuins cannot function in the same way that even the most sophisticated repair robot cannot work without electricity. Between the ages of 40 and 70, NAD+ levels in human tissue decline by approximately 50%, according to research from Washington University in St. Louis. This decline is a primary reason DNA repair becomes so inefficient with age. The most evidence-supported dietary sources of NAD+ precursors are niacin-rich foods, specifically turkey, chicken, tuna, and nutritional yeast, and a compound called nicotinamide riboside found in trace amounts in milk and certain yeasts. But the most accessible and potent dietary strategy for raising NAD+ is through a different pathway entirely: Consuming foods rich in tryptophan and amino acid found in eggs, pumpkin seeds, and turkey, alongside regular moderate exercise and intermittent fasting periods of 14 to 16 hours, which have been shown by researchers at the Salk Institute to increase NAD+ biosynthesis by up to 50% independent of dietary intake. Research from the University of New South Wales found that combining dietary tryptophan with regular fasting periods increased DNA repair enzyme activity by 44% in adults over 65 over a 12-week period. My patient Raymond, 69, from Baltimore, Maryland, came to me following a routine physical during which his physician noted elevated inflammatory markers and what his doctor vaguely described as signs of accelerated cellular aging in his blood work. Raymond was not sick in any conventional sense, but his biological markers painted a picture of a man whose internal repair systems were significantly underperforming. Raymond was a night shift worker who ate irregularly, rarely fasted, and consumed almost no tryptophan-rich or NAD+ supporting foods. We restructured his eating window to a 12-hour daily fast, simply moving his last meal to 7 p.m. and his first meal to 7 a.m., added eggs each morning, pumpkin seeds as a daily snack, and a small amount of nutritional yeast on his lunch. At 16 weeks, his inflammatory markers had dropped measurably, and a follow-up cellular aging assessment showed DNA damage markers that his physician described as significantly improved for someone his age. Practical implementation. Begin with a 12-hour overnight fast-finish dinner by 7 p.m., eat breakfast at 7 a.m. This alone meaningfully increases NAD+ biosynthesis. Add 2 whole eggs each morning for tryptophan and B vitamins. Incorporate 1 tablespoon of nutritional yeast daily. It has a mild, cheesy flavor and dissolves easily into soups, salads, or scrambled eggs. For synergy, pair your NAD+ supporting foods with resveratrol-containing foods, specifically red grapes, blueberries, or dark chocolate above 85% cacao, because resveratrol directly activates sirtuin enzymes, the DNA repair proteins that NAD+ powers, creating a compounding activation effect. Research from Harvard's Paul Glenn Center for Biology of Aging found this combination increased sirtuin activity by 37% above either nutrient alone. Number 4, and this intervention operates at a level of biological organization so fundamental that it affects every other system on this list simultaneously: the restoration of mitochondrial health and the triggering of mitochondrial biogenesis, the creation of brand new cellular energy engines as a primary mechanism of biological age reversal. You have heard me discuss mitochondria in previous videos, but today I want to go deeper because the connection between mitochondrial health and DNA aging is more intimate than most people realize. Mitochondria, the energy-producing organelles inside every cell, have their own separate DNA, distinct from the DNA in your cell's nucleus. Mitochondrial DNA is far more vulnerable to damage than nuclear DNA because it lacks the protective protein coating called histones and sits in close proximity to the reactive oxygen molecules that mitochondria produce as a byproduct of energy generation. Think of it like working in a factory that produces sparks. The workers closest to the furnace get burned most often. By the age of 65, mitochondrial DNA mutation rates are approximately 4 to 5 times higher than in young adults, and mitochondrial efficiency, the amount of usable energy produced per unit of cellular fuel, has declined by 30 to 40%, according to research from the Buck Institute for Research on Aging. This mitochondrial dysfunction creates a cascade that drives virtually every visible and invisible sign of aging: fatigue, cognitive slowing, muscle weakness, immune decline, and accelerated telomere shortening caused by the oxidative stress that dysfunctional mitochondria produce in excess. The most powerful non-pharmaceutical trigger for mitochondrial biogenesis, the creation of new, healthy mitochondria to replace damaged ones, is a molecule called PGC1 alpha, which functions as the master switch for mitochondrial generation. Think of PGC1 alpha as the construction permit that allows your body to build new energy plants. Several specific dietary compounds activate this switch, with evidence comparable in some studies to pharmaceutical interventions. Urolithin A, a compound produced when gut bacteria metabolize a substance called L-agitanin, found in pomegranates, walnuts, and raspberries, was shown in a study from the Amazentis Research Group published in Nature Metabolism to increase mitochondrial function in muscle tissue by 42% in adults over 65 after 4 months of consistent consumption. This same study documented improved muscle endurance and reduced cellular aging markers, and it was a randomized controlled trial, the gold standard of medical evidence. Quercetin, a polyphenol found in red onions, capers, and apple skins, has been shown in research from the Mayo Clinic to selectively clear senescent cells, the zombie cells I described earlier, while simultaneously activating PGC-1 alpha and stimulating mitochondrial biogenesis. A 2018 clinical trial found that quercetin supplementation in adults over 65 reduced senescent cell burden by 35% and improved physical function scores significantly over 11 days of concentrated dosing. My patient Evelyn, 74, from Tucson, Arizona, described her primary experience of aging as running on an empty battery by 2 in the afternoon. Evelyn slept adequately, had no thyroid dysfunction, and had been told repeatedly that her fatigue was simply a normal part of aging. When we assessed her dietary pattern, she consumed virtually none of the mitochondria-supporting foods, no pomegranate, no walnuts, no quercetin-rich vegetables, no urolithin precursors of any kind. We introduced a daily smoothie containing pomegranate juice, raspberries, and walnuts, added a daily red onion to her salads, and incorporated apple slices with the skin intact as her afternoon snack. Within 6 weeks, Evelyn contacted my office to tell me something she described as almost embarrassing and how simple it seems now. She had energy in the afternoon for the first time in 4 years. Within 4 months, she had resumed a watercolor painting practice she had abandoned due to fatigue 2 years prior. Practical daily implementation: 1 cup of pomegranate juice or half a pomegranate daily provides the L-agitanin substrate for urolithin A production, though individual conversion depends on gut microbiome composition. 1/4 cup of walnuts daily adds additional L-agitanin and alpha-linolenic acid. 1 cup of raspberries provides both L-agitanin and quercetin. 1 medium red onion, raw or lightly cooked, delivers a substantial quercetin dose. For synergy, combine these foods with consistent, moderate aerobic exercise, 30 minutes of walking or cycling, because exercise is the most potent known activator of PGC-1 alpha, and research from the University of Copenhagen found that the combination of dietary polyphenols and aerobic exercise increased mitochondrial biogenesis by 61% more than exercise alone, creating a compounding regenerative effect that neither intervention achieves independently. Right here, I want to pause because the top 3 items on this countdown represent the frontier of longevity science findings so significant that they have reshaped how researchers think about aging at the most fundamental level. If you are finding this valuable, please hit the subscribe button right now and tap that like button. It takes 3 seconds and helps me continue bringing this caliber of evidence-based content to every person over 60 who deserves to have it. Now the top 3. Number 3. And this is the intervention that Harvard Medical School researcher Dr. David Sinclair has described as among the most promising natural longevity mechanisms identified in the past 2 decades. And it involves a cellular recycling process that cleans your DNA, clears damaged proteins, and resets cellular age in ways that no supplement currently on the market can fully replicate: the strategic activation of autophagy through specific dietary patterns and compounds. Autophagy, as I've explained before, is your body's cellular self-cleaning system, the maintenance crew that moves through each cell, identifies and removes damaged components, and recycles the raw materials into new, functional cellular machinery. But what most content about autophagy never explains is its direct relationship to DNA integrity. When cellular debris, damaged proteins, dysfunctional organelles, oxidized lipids accumulates inside cells, it creates a toxic microenvironment that accelerates DNA damage and impairs DNA repair enzymes. Autophagy clears this toxic environment and restores the cellular conditions under which DNA repair can operate at full efficiency. Restoring autophagy does not just slow aging. It reactivates the machinery that repairs aging's primary damage. After the age of 65, autophagy activity declines by an estimated 40 to 50%. Research from the University of Texas Southwestern Medical Center found that this decline is a major driver of the senescent cell accumulation and chronic inflammation, what longevity researchers call inflammaging, that characterizes aging at the cellular level. Spermidine, a naturally occurring compound found in extraordinarily high concentrations in aged cheese, wheat germ, mushrooms, and legumes, is the most potent known dietary activator of autophagy. A landmark study from the University of Graz in Austria, published in Nature Medicine, found that higher dietary spermidine intake in adults over 60 was associated with a 25% reduction in all-cause mortality and significantly reduced cognitive decline rates over a 20-year observation period. 25% reduction in all-cause mortality from a dietary compound found in food you can buy at any grocery store. Mechanically, spermidine activates autophagy by inhibiting a protein called EP300, which functions as a brake on the cellular cleaning process. Think of EP300 as the manager who keeps telling the maintenance crew they can take a break. Spermidine fires that manager. The maintenance crew goes back to work, clearing cellular debris, restoring DNA repair environments, and removing the senescent cells that were releasing inflammatory chemicals into surrounding tissue. My patient George, 78, from Boston, Massachusetts, had been following every mainstream longevity recommendation, caloric restriction, exercise, stress reduction for 3 years with modest results. His biological age assessments using DNA methylation testing had improved only marginally. When we reviewed his diet, his spermidine intake was virtually zero no-aged cheese, no wheat germ, minimal legumes. We added 2 tablespoons of wheat germ to his morning oatmeal, incorporated aged Parmesan into his daily lunch, and increased his legume consumption to daily servings. At 12 weeks, his DNA methylation biological age assessment showed a shift that genuinely surprised both of us. His biological age marker had moved by 1.8 years in the younger direction in 3 months. George called me from his kitchen to tell me he had just done 20 push-ups for the first time in a decade. Practical implementation. Wheat germ is the single highest dietary source of spermidine at approximately 24 milligrams per 100 grams. 2 tablespoons daily in oatmeal, yogurt, or smoothies provides a meaningful dose. Aged hard cheeses, particularly Parmesan and aged cheddar, are the second highest source. Lentils, chickpeas, and soybeans provide substantial spermidine alongside fiber that supports the gut microbiome pathways that further amplify autophagy. For synergy, combine spermidine-rich foods with a 16-hour overnight fast 3 to 4 times per week, as fasting activates autophagy through a separate AMPK pathway, and research from the Longevity Institute at the University of Southern California found the combination of dietary spermidine and intermittent fasting increased autophagy activity by 78% above either intervention alone, nearly doubling the cellular cleaning effect. Number 2, and this is the item that brings together genetics, epigenetics, and the most exciting frontier of aging science in a single accessible intervention, the dietary modulation of epigenetic markers, the molecular switches that turn genes on and off to actively reprogram the biological age of your cells. Epigenetics is the study of how your genes are expressed without changing the underlying DNA sequence. Think of your DNA as the complete musical score for a symphony. Epigenetics is the conductor, deciding which instruments play, how loudly, and for how long. As you age, these epigenetic patterns become dysregulated. Genes that should be silenced, including inflammatory genes, cell division genes, and stress response genes, become active, while genes that should be expressed, including DNA repair genes, antioxidant production genes, and regenerative pathway genes, become silenced. This epigenetic dysregulation is now understood to be one of the primary clocks measuring biological aging. And it is what the DNA methylation clock, the biological age test I mentioned at the beginning of this video, actually measures. The revolutionary insight of the past decade is that epigenetic patterns are not fixed. They respond to diet, to behavior, to environment, and certain specific dietary compounds have been shown to directly and measurably shift epigenetic patterns toward a younger biological state. The most evidence supported of these compounds is sulforaphane, a molecule produced when cruciferous vegetables like broccoli, Brussels sprouts, and broccoli sprouts are chewed or chopped. The chopping or chewing triggers an enzymatic reaction that converts a precursor compound called glucoraphanin into sulforaphane, the active molecule. Sulforaphane activates a transcription factor called NRF2. Think of NRF2 as the master switch for your body's entire endogenous antioxidant and detoxification system. When NRF2 is active, it turns on over 200 protective genes simultaneously, including genes for glutathione production, your body's most powerful internal antioxidant DNA repair enzyme production, anti-inflammatory cytokine regulation, and mitochondrial protection. A study from Johns Hopkins University, where sulforaphane was originally characterized, found that sulforaphane supplementation in adults over 65 increased NRF2 activity by 54% and measurably shifted DNA methylation patterns toward younger biological profiles over 12 weeks. A 2021 trial from the University of California, Los Angeles, specifically measured DNA methylation biological age in adults averaging 67 years old, who followed a comprehensive epigenetic intervention, including sulforaphane-rich foods, alongside other dietary and lifestyle factors. The results showed an average biological age reversal of 3.23 years in 8 weeks, the study I referenced in the opening of this video. Sulforaphane-containing foods were among the highest impact dietary components in that intervention. My patient Diana, 66, from Seattle, Washington, was a retired scientist who understood the epigenetics research conceptually, but had never systematically implemented it. Diana's biological age assessment placed her 4.1 years older than her chronological age, a finding that disturbed her deeply given her attention to conventional health practices. Diana's diet contained almost no cruciferous vegetables. She disliked broccoli and had avoided it for decades. We found a solution: broccoli sprouts, which contain 50 to 100 times more glucoraphanin than mature broccoli, and have a milder, slightly peppery flavor that Diana found acceptable. 1 tablespoon of fresh broccoli sprouts on her salad daily. Within 16 weeks, Diana's biological age reassessment showed a shift of 2.7 years in the younger direction. She forwarded me the results with a single line: "I owe you a broccoli sprout apology." Practical implementation. The most important preparation detail is that sulforaphane is destroyed by cooking above 70 degrees Celsius. Raw, or very lightly steamed cruciferous vegetables are the target. Broccoli sprouts are the highest yield, lowest volume option, 1 tablespoon daily on salads or in sandwiches. Mature broccoli, raw or briefly blanched, 3 to 4 times per week. Brussels sprouts, halved and eaten with minimal cooking. For the maximum synergy enhancement, add mustard powder or mustard seeds to any cooked cruciferous vegetables. Mustard contains an enzyme called myrosinase that compensates for the myrosinase destroyed by cooking, restoring sulforaphane production from cooked broccoli to approximately 70% of the raw level. Research from the University of Illinois found that adding mustard to cooked broccoli increased sulforaphane bioavailability by over 400% compared to cooked broccoli eaten alone. And now, number 1, the intervention that I believe represents the most accessible, the most profoundly documented, and the most urgently underutilized tool for DNA repair and biological age reversal available to every person over 60 watching this video: the restoration and optimization of the gut microbiome as the primary control system for epigenetic aging, immune function, DNA integrity, and systemic inflammation simultaneously. I told you at the beginning that number 1 involves a mechanism so fundamental to your DNA that pharmaceutical companies have spent billions trying to replicate it artificially. Here's what I meant. The 38 trillion microorganisms living in your gut are not passengers. They are active participants in your biology at the most fundamental level. They produce short-chain fatty acids that directly regulate epigenetic gene expression throughout your entire body. They synthesize B vitamins, including folate and B12, that are essential cofactors for DNA methylation, the very process that controls your biological age clock. They regulate the systemic inflammatory environment that either protects or degrades your telomeres. They modulate the immune surveillance systems that identify and eliminate senescent cells. They produce urolithin A, the mitochondrial biogenesis compound we discussed earlier. They influence NAD+ biosynthesis. In short, your gut microbiome is the biological infrastructure upon which virtually every other longevity mechanism in this video depends.